focused on that are trying to identify EB patients and trying to develop centers that can greatly help the patients. So focus on benefiting the patients. We did questionnaires to 216 patients, and over 68% responded, half male, half female. And our data showed that when we looked at the scores, that patients described that itchiness was the most bothersome EB complication, and the score we had was more bothersome than pain. And so we thought this was a very important point. Next slide, please. Other thing that they noticed was pruritus was more frequent at bedtime, interfered with sleep, was aggravated by dry skin, wounds, stress, heat. And all these things are things that we had heard before. All these things are things that we thought we knew, but doing clinical research in the future, we need to document these things, and this survey was able to do that. Next slide, please. The next patient, I think we skipped a slide there. If you go back, there we go. From the same survey that was used, we asked patients exactly what kind of treatments they did. And so you can see ointments, lotions, hydroxyzine, creams are frequently used. Next slide, please. But the treatments and the patient's perception of the treatment, basically all of the treatments were considered to only relieve itch a little. And this is something that many of you know. This is something that we know. But it's important to document these things, because as you try to develop new styles, new studies in the future, we'll need to understand what's there. And so currently I know there are some randomized trials for better therapies that are in development. There are some new medications that are out there. And so there is an effort to try to develop randomized trials. So hopefully within a year or two, we'll be able to report back on some of that. Next slide, please. So there's another study trying to look at how to evaluate if someone has EB, how severe their EB is. This is a group of pediatric dermatologists across North America and Canada who are looking at what is an effective method we can use to evaluate EB. And so what they did, they developed a tool, and then they examined 21 consecutive patients, 2 different sites to try to identify how it worked. Next slide, please. Now realize that as therapies are being developed, the Food Administration, both in the United States and Europe and Japan and all over the world, is going to say we need to know what's effective. What is the efficacy of the things that we're doing? This is a kind of a trial tool that we need to use. And so what they identified, they looked at the skin, the mucosa, the different organs of the body that are involved, asked patients and had systems to look at pain, itch, sleep, mood, lots of activities of daily living. Next slide, please. What to show is that this tool called iScore EB seems to be sensitive and differentiating between the EB types and across the clinical spectrum. So the hope is that with further validation, we'll be able to say that patient A was given such a treatment, and patient showed a quantitative evaluation of this improvement or lack of improvement be satisfactory for the FDA. Next slide, please. So one of the areas of controversy in EB care is what's the best wound dressings? What are the ways that we help patients with the wound dressings, and what are the things we could do? Should we be giving IV stem cells? Should we be using amnion or chorion? And so a study of an 11-month-old baby with junctional EB who we 5 infusions of cells, and these cells were removed from the products of multiple babies, and so received amniotic membranes that were tied to wounds. The doctors were able to document that the wounds improved, but then after time the wounds were back to where they were. But next slide, please. What they all documented is that these patients being exposed to all these different cells from different people developed many antibodies to proteins that would be in you or I, but would not necessarily be in this child with JEB. So the amount of immune response that this child showed to all these foreign membranes and foreign cells was greater than what they expected. And so when cultured, they're often cultured in a media that contains calf serum from a fetus, from a calf, and it turns out that this patient also developed antibodies to that. So this sort of lends a warning to some of the stem cell so people were trying to develop that eventually we may develop antibodies and limit the ability of people to receive different treatments. Next slide, please. Another specific area of research that people were focusing on is how does EB, next slide please, how does EB affect your lives? How does EB make a difference? And if we don't have effective therapies, or as we develop effective therapies, what are the things we can do in the meantime to try to help families and basically try to develop communities that can help families? And this was a study that was done by Deborah Ireland, where they admitted that the primary needs for patients, such as bandages, information, and the pain and suffering, was just really inadequate in the surveyed population. Next slide, please. They felt that this paper may help develop within their country public policy changes that would prioritize helping patients with EB. Citizens become aware and enable systems that would be seamless and sustainable through collective action. So one of the outcomes of some of these in foreign countries and what we're trying to develop in the United States, EB centers that have the physical therapist, to have the hematologist, have the other physicians who are to help with EB, and this is a document that supports that. Slide, please. Another thing that's a big impact that's going to have for EB care in the future is what's called whole-exome sequencing. So many of you out there have had genetic testing done to evaluate how, what is the specific mutations, so, what's the specific diagnosis. But what in the last 3 or 4 years, there are whole new technologies that very rapidly can evaluate someone's genome and identify not just, one gene for EB, but look for all the genes at the same time. Next slide, please. And so current sequence is basically looking like one, walks way down the genes that are involved. The technique that involves looking at multiple mutations at the same time. And in this study, they took 9 patients with EB who they were not able to identify which type of EB had based upon genetics. And using whole-exome sequencing, they were able to identify all 9. And what this means in the future, as we get into complicated clinical trials, we'll probably know the genetic diagnosis on each child with EB, as would be other skin diseases, because the therapies may become specific based upon the genetic diagnosis. And often this is a requirement the Food and Drug Administration has. But what this paper is emphasizing is the cost of doing that is going down rapidly. And in Europe, because they have a national health care, almost everybody gets genetic testing. In the States, that doesn't happen because many insurance companies refuse to pay for it. But future testing may become less expensive, may become faster. It may really be helpful to identify and improve therapies by knowing specifically the mutations. Next slide. Areas of a lot of the research articles that were published were in cell-based therapies, and there's many different cell-based therapies that are being developed, and I'll walk through some of those. Why don't we go to the next slide? So you're aware that some centers are doing bone marrow transplant. So there were a lot of different studies trying to look at what are the things you can do to make bone marrow transplant work? What are the things you can do to have it when it does work even better? So there are different growth factors and chemicals that interfere with the cells and the cell's growth. So this was an example where they used a specific type of platelet-derived growth factor receptor, positive stem cells, and they will show that when these stem cells were used in a mouse, the mouse who was not making type 7 collagen now could make type 7 collagen. To realize when we look at the mouse studies is basically the mice that are used for these kind of studies are sort of like identical twins, where one mouse has EB and the other mouse doesn't. So the only difference is that the fact that it has the effect in type 7 collagen. So all these results is one example of how people are trying to look at better ways to doing some of the things we're doing. And the next slide is similar. This would be, and this, so the next slide, please. This was an example where these doctors were using a different type of a stem cell, which they called unrestricted somatic stem cells, USSCs. And then next slide, and they were able to show that in these mouse models that they, by injecting these cells into the mouse's liver, that the mouse with recessive dystrophic EB would live a few days longer. So another example of trying to find better ways of doing some of the things that we're doing currently. Next slide, please. Another looking at this, and again, these are all trying to look at this model and to say what are the things we can do to try to make the mouse model better, and then attribute that to humans. This is an example where they were doing multiple different chemicals exposing the cells to try to stimulate proteins which may make more type 7 collagen. Now, these studies in mice may eventually help guide the studies in humans, but a lot of the EB studies that are done looking at cell therapy has been done in mice. Next slide, please. So the next sequence and the last sequence that I'd like to focus on is the area of modified cells. So this would be an area where we take someone's skin cells or whatever cell and genetically correct it and then give it back to them. And there's been a lot of progress in this in the last 5 years. Next slide, please. So realize that 7 years ago, we heard a lot, or even more than that, a lot of complaints about using embryonic stem cells. And so the Bush administration and then followed by the Obama administration, there's been this fight back and forth. You haven't heard much about that fight in the last 3 or 4 years. And because the embryonic stem cells are not used as much now. And so what happens with an embryonic stem cell is what's shown in this diagram is you can, as it divides into another cell, those at the bottom could be twins. They could divide it again, and they could be quadruplets. That's what happens with an embryonic stem cell. Next slide, please. And take those cells at the bottom, and they could go into becoming brain or heart or kidney or skin. Slide, please. But there's a type of cell that's out there now that's called the IPScs cell, or inducible pluripotent stem cell. And that stem cell, basically, you can make in the lab. So what we do is we biopsy the skin, and we can take either the outer layer skins, the keratinocytes, or the inner layer skin cells, the fibroblasts, and we can grow them, and we can change them into inducible pluripotent stem cells. Next slide, please. And if we take an adult cell or a child cell, we force them to become just like they would then as a stem cell in the embryo. So it's a way to push the cells back many, many years, and then you're able to take those cells and convert them to skin or to heart. And skin and heart are currently the big areas that this type of therapy is focused on currently. Next slide. Another thing, as I review the next studies, attorneys need to be aware of is what's called revertant mosaicism. It turns out there are some people with EB who have areas on their skin that never seem to blister, areas that chronically blister, and you have other areas that don't seem to blister. How often that is, how many patients have that, it's not clear, but sometimes what will happen is on recessive disease where you have two mutations, sometimes a third mutation occurs which sort of counteracts one of the other two mutations, and the patient's skin becomes like that of one of the parents, which has just one mutation. So this is called revertant. Basically, you have a mixture of disease cells and a mixture of cells that had another mutation that are not as diseased or maybe mildly effective or not affected at all by that specific disease. It happens in EB, it happens in other diseases, but that's important to understand the term revertant mosaicism. Next slide, please. So this study where they took a patient with recessive dystrophy, and they took keratinocytes and reprogrammed them into inducible pluripotent stem cells. So this had been done before. This had been done in 2011. This study was published in 2014. But what these authors did also, besides making these cells go into looking like skin cells, these also go into looking like blood cells. It's clear whether these cells would grow as skin, but they had the characteristic of skin. That other additional information wasn't in this. Next slide, please. This is a study looking at the cells of a patient with junctional EB. And what we're able to do was a process called homologous recombination. You have a recessive disease. You have two chromosomes that have mutations on it. Think of the gene. Let's say the gene is a yardstick. It's 36 inches long. One mutation may be at 10 inches, and the other mutation may be at 25 inches. So one parent carries the 10-inch mutation, the other one carries a 25-inch mutation, and then the baby has both mutations, and so they have the disease. But there are times when you're able to take the mutation, either the 10 or the 25, and switch the chromosomes so that you end up with one chromosome that has a mutation at 10 and 25, and the other one that doesn't have any mutation at all. And that's called homologous recombination. You basically take the patient's own DNA, but you switch it. You give part of it to one chromosome and move it to the other chromosome. Next slide, please. These authors took a virus, and they were able to force these cells to develop homologous recombination. And then they were able to identify the cells that are now corrected as they grew better, and they stuck on the culture plate, and then able to take these cells and grow them into skin. So it's an example where they took a disease, they used the inducible pluripotent stem cells, and they were able to then convert these cells and grow up the cells that are more acting like normal. And so there's a chance that these cells may eventually be able to be grafted to the patient. Next slide, please. This is another study using IPS cells, inducible pluripotent stem cells, but this group were able to identify specific IPS cells that had less frequent mutations, which may lead to skin cancer. So we know that with recessive dystrophic that over time we see skin cancers, and there's a chance that you could see and try to make cells that would likely induced into IPS cells, and yet they may also have a tendency to go towards cancer. So this is a group that was able to identify the cells which were less likely to become cancer, and then they were able to take these cells, grow them up, and plant them on a mouse. There's a mouse that accepts human skin, grow them, and they were able to document that you could take like a fibroblast, made into a inducible pluripotent stem cell, look at stem cells and say this one has a high risk of cancer, this one has a low risk of cancer. The one that has a low risk, let's convert it to skin, and with the hopes of eventually giving this back to the patient. So there's a major advancement in the inducible pluripotent stem cells. The next slide is also, the one you have on now is perfect. It's also one that looked at the same thing, but then they took the revertants and they were able to generate IPS cells in type 17 collagen. So this is a junctional EB patient. And if to grow these cells, make them IPS cells, use the reverse ones, the ones that have the mutation, and then grow up the skin. So this was an also very similar but very positive response. Next slide, please. And this was a study that was done in mice. So, we're able to take mice who had RDEB. They were able to genetically make IPS cells and tried to make keratinocytes, but they couldn't. But they were able to make fibroblasts, which were genetically corrected, and then they were able to show that the fibroblasts injected back into the mouse with RDEB, they were able to look like they had improvement in the skin. So that's the next slide would be questions. And so what I had to show here was that there are some very simple studies like the salt water in the bath that may have immediate impact and immediate improvement for some people, while there are studies that are much more complex that may have many years from now potential to have benefit. But the good news is there's a lot of people out there working on EB, and there's a lot of things that have been published just in the last year. So I'd be happy to answer questions if people have questions. This, what you'll do is you'll type them in. I'd like to focus on questions related to some of the studies that I presented.
You haven't been able to find the chat function. It's at the top of the screen where it says Shift Home or Desktop. You can look for it there, hit the chat, and you should be able to type your question in, and you can direct it towards Al Lane, and he should be able to start seeing them. Al, are you any questions in?
I don't have questions yet. But see you come in? Yep. Would you just send them to everybody? Could they do that? Or they? It's on Article 13. Can the IPS? So here we go. It's on Article 13. Can the IPS be generated for mutation for JEB so the trachea? So this is the AAV article. So that's an excellent question. So there are different types of skin that we have on our body. We have mucous membranes, which are in the vagina, the rectum. We have a different type of like a membrane over the eye and the cornea. We have different skin cells that are different on the palms and the soles, different than the arms or the legs or the trunk. And so this question about the trach, can you go for the trachea? That's an excellent question, but the answer we don't know yet, because some of these cells have what's called donor dominance. In other words, the cells that are in the trachea are influenced by the tissue underneath those cells, so the tissue of the trachea or the tissue of the esophagus. And how you manipulate those changes isn't always clear. And so that's the step number 5, where just initially just grafting to the normal skin would be step number 1. It's a much more complicated thing, but we're thinking about exactly that at this time. Okay. So the question, protein therapy. And so what I tried to do in this study was just to go with things that are published, because the information that's published is public knowledge. There are a lot of different centers that are working on different approaches. So I talked about space therapy. We have a program in transfer at Stanford where we're trying to take patient cells and genetically change them by putting in the correct gene with the virus. There are other studies that are being done where in types of EBs you're missing specific proteins, and there are efforts to try to put those specific proteins into the cells. but currently haven't been any studies that have been published on these areas in the last year and a half. As far as the protein therapy, there are some studies that were published many years ago in mice, but there was nothing in the last year and a half, so that's why I didn't include that. And there's some other questions that are looking, what are we doing at Stanford? Are we recruiting? So anything, the one way that people can find out about what's going on is a program called clinicaltrials.gov. Just those two words together, .gov, and search that. And then when you get into that, you can just type in Epidermolysis Bullosa. And that lists all the studies that are going on in the United States. If someone has a study that's open or looking for subjects, that's where it would be. And so there's some questions about how soon is Stanford going to move to children's and our gene transfer. Again, Stanford has a very structured rule for us. We're not allowed to talk about our research until it's been published in a peer-reviewed journal, because the feeling is we don't want to give out information that may not be proved to be accurate later. I really can't give those results. There's a question about glaucoma. That really wasn't something I covered. There's a cord blood banking of cord blood for a future child. Again, there was nothing published in the last year about that. There's a question about revertant mosaicism. So revertant mosaicism is, how can you test for that? Basically, there are a couple of publications that I mentioned. I'm not aware of any trial that's going on clinicaltrials.gov looking for patients with revertant mosaicism, but these are usually patients who have one area that just never seems to blister. Turns out it's more common with the junctional EB, collagen 17, where I've heard that most of the patients have an area like that. It's less common with the recessive dystrophy. And so right now, again, clinicaltrials.gov is the best place to keep watching that. And I look at that probably every couple of months, like spring, summer, winter, and fall, to see what studies are going on. Question about local studies, East Coast. Again, clinicaltrials.gov is where a study should be doing. Question about modified cells in humans. All those things have to go through the FDA, and I know that many centers are talking to the FDA. Again, that's very private communications. I don't have access to that. But again, these things that have been published, realize that they publish because they've been reviewed, they've been shown to be accurate by other investigators reviewing the articles, and the hope is some of those things will become in the future. Will kids with revert mosaicism still be candidates for treatment someday? Absolutely. It turns out there may be some advantages to having revert mosaicism, and that's why so many centers are trying to look at that now. What therapies do you foresee as a future cure treatment for RDEB? I think their combination, genetically corrected grafts. I think what's going to happen is there may be a combination of therapies. Ideally, you like to be able to infuse like the protein or infuse cells, which then would go to the skin, the esophagus, and the mucous membranes. But all these things complicated. Some methods that we use to grow the cells may increase the risk of developing a cancer. So all these things are aggressively looked at, and when we'll have a result, I'm not sure. Question: dominant mutations, cell corrections. Yes, the IPS cell method is effective for dominant. The advantage of it is you can genetically take a cell with homologous recombination, and you can select cells that you've been able to take the mutation out, and so this is important. Question about pool salts will dry out the skin. If so, how do you stop it? So what this study suggested is that it doesn't dry out the skin. And as we saw in our study with pruritis, is many patients are using lubrications or different ointments. As far as the pruritis, there's lots being used, but people didn't think that they were doing a great job. But to my knowledge, the pool salts not dry out the skin, and that wasn't a side effect that they saw in that study. Treatment for one form or other forms, specific forms. Basically, a lot of the research that's currently being done that I mentioned were focused on recessive dystrophy. And reasons for that is because there's a mouse model for that disease. And investigators are able to do things in mice that we can't do in humans because of safety factors. So there's also, I know, a mouse model for junctional EB. There are mouse models for AAV simplex. And so a lot of these things that, again, that are being done, they just weren't published in the last year. And the things that you'll have is that often it takes several years of research to have a publication. But I know that people specifically looking on dominant dystrophic, and they're looking at the dominant simplex group. I'm not sure. So does severity or the type of the disease have effect on mutation? There are a lot of studies trying to look at specific mutations in the different types of EB and saying which would be more severe in one child than another. And it is not very clear that mutations can identify severity of disease. Families where we have more than one child with EB, we find that one child is more severely affected than others. So besides the genetics, it could be the activity of the child, it could be secondary infections, it could be multiple other things. So currently, we can't say that if you have one genetic mutation, it's going to be worse than another. The question is Stanford working on revertant mosaicism? We had a study that we were looking at revertant mosaicism, but that's not a focus currently. But definitely, we're looking for patients with revertant mosaicism. We see them in the clinic, and we would refer those patients to other centers that might be more focused. Question: bleach and pool salts? Yes, people are using bleach baths with the pool salts. Mention about what's called messenger RNA studies. Nothing published in the last year. I know there are multiple groups looking at that, especially trying to look at the dominant. About me, working on EB for a long time. How do I feel about meaningful treatments? I have recently retired from Stanford, but I'm still involved in some of the activities that are going on. And I think that the hope that I have for the next 5 years is that things are going to go much faster than they were in the previous 20. One of the big changes that's occurred in the last several years is the Food and Drug Administration has changed the rules and the funding systems, the way they look at orphan diseases. And EB is an orphan disease in the United States. That means there's less than 200,000 patients with it. And there are many companies that may have a financial advantage to getting involved with orphan diseases. So before, as a research center, we collect any funds we can. If industry gets involved, they're often better funded. They have a profit motive, but they're often better funded. So I think things will move a little faster. And then I think we're just about at time.
Yeah. I'm going to go ahead and interrupt you. Thank you, everyone, on behalf of EB Research Partnership, for joining us today and listening to Dr. Lane's presentation on where research is in the last year. We will be having another webinar on July 14th, and I'm going to give Dr. Lane another opportunity to take a few more questions. I just wanted to say another quick thank you to our sponsors, FiberCell Science and McKesson Patient Care Solutions. We thank you so much for making today possible. If you'd like to, you can take a few more questions.
Sure, a few more questions. So the question is, So patients who don't make type 7 collagen or even collagen 17 or all the proteins in the basement membrane that can be associated with EB, there's a chance that if you correct the disease, could the patient develop antibodies to those proteins? And the answer is yes, definitely. But the other things that has been running parallel research, which is bone marrow transplant research, kidney transplant research, heart transplant research, liver and lung transplant research. So in those situations, they're giving a whole different organ. They're not making one change on one protein. So there's been a lot that's been done in the last 10 or 20 years on organ transplants that can be very helpful to us as we get into more complex studies with EB and where we're replacing proteins. So other research not related to EB that may have a great benefit to those patients definitely will be in the future. Questions about reducing inflammation and granulation of larynx. Again, nothing published in the last year and a half that I saw on that. Some thank yous is, some questions we're getting here. I'd like to emphasize again, I did, when I had the opportunity to do this talk, to just see the large number of publications during every year related to EB. I think that's phenomenal. I know that internationally, that communities in Europe and Japan and Asia are starting to work together. South America, there's a lot of collaboration that's going on because this disease is so severe. So I think there's a great hope for the future. And I think that's the last question.
Thank you, everyone. Thank you, Dr. Lane. It's been a privilege.
Thank you. And good luck to everybody, and there's hope for the future.